I got here 86. Everything work? Oh, I can't tell if everything's working. Can anybody hear me?
Yeah,
Yeah, we can hear you.
Yep.
Very good. So let's see here. I think we were here in smooth muscle somewhere. Yeah. And if you're wondering about the exam, we'll go over the exam and recitation. If you haven't gotten your email back, check your email. It should be in there. Average was about 80% or something like that. I'm going to add a point to every one of them because I don't know, nobody got 100. There were lots of 49s and lots of 48s. So I'm going to add a point to each one. And I'll go over on Thursday in recitation. Just as a little blast from the past here, thinking about some of that content, as we go through smooth muscle, you'll notice a lot of similarities between how smooth muscle works and how skeletal muscle works. A lot of commonalities in terms of the biochemistry and some also very important differences that really illustrate how weird skeletal muscle is, how it has a very different philosophy in terms of what it's intended to do relative to smooth muscle. And again, I think we were right about in here. Smooth muscle is never loses its individual identity. It retains the ability to undergo mitosis. There are spectacular examples of smooth muscle that proliferates and then atrophies. The mammalian uterus being perhaps the best of all, greatly increase in size. It has a uterine muscle has a very different task than a simple locomotion. And the smooth muscle is found all over the place. Any kind of tube that is a cross section, if you had in cross section some tissue that looked like this in cross section, odds are there's going to be smooth muscle on the walls of it. And that will allow the tube to alter the size of its lumen, that is the opening in the middle, and thereby greatly change the flow of what's ever going through that tube, be it a food bolus in the digestive system or sperm inside of vas deferens or blood inside a blood vessel. If you change the size of that lumen dramatically by contracting the smooth muscle around here, you will very much change how that stuff proceeds through that tube. Clear all bad drawings. So it can rearrange in different in ways. Multi-unitary has a lot of nerves attached to it, versus unitary is one that it functions as a one unit, and you essentially you touch off a part of a unitary smooth muscle and the whole thing tends to eventually contract. Versus multi-unitary is much more, in terms of its innervation and coordination, is a bit more like skeletal muscle in that it's going to contract in concert because it has nerve endings attached to all of the cells. So they kind of look like this. There we go. There we go. Oh, where did they go? Did I delete a slide? Oh, that's what I wanted to see. So here's the multi-unitary. The entire group of smooth muscles all have a part of a neuron talking to them, versus a unitary might have just a portion of it innervated, and then eventually they all talk to each other as the cells themselves transmit the information. We're contracting now. To go back a slide now. Oh, by the way, I reposted these slides because one of the slides further on down here was just a piece of crap. Came from a textbook and used it for 15 years and never been terribly happy with it, but it had all the words, and so, but I fixed it. I think it's a little easier to read now. So if you want to download them again, you can. Smooth muscle is different if you're looking within a cell. The arrangement of the sarcomeres is not such a regular thing as it is in skeletal muscle. Skeletal muscle, everything is regimented. All the myofibrils, all the myofilaments within them are all lined up such the sarcomeres are just in rank and file. Not so inside of smooth muscle. And what that means, of course, is that the force that they produce is going to be in more directions. So if you're looking within a smooth muscle and these red lines are the actual sarcomeres and the myofilaments, then when they contract, some of them are going to go going this way and some of them are going to be going this way, and so on and so on and so on. The downside, of course, is that they're not all together in terms of the direction of the force they're producing. So, some of them are going this way and some are going this way. The upside is because they're not regimented, that means when they contract, they're all going in slightly different directions and they don't get in each other's way. It's the difference between a shock absorber that everything, kind of hard to explain, if you just imagine a spring going like this versus, say, a Chinese finger puzzle. Do they still make those stupid things? The Chinese finger puzzle where you stick your fingers in and then you try to pull them apart and they get stuck? Obviously, I can't do it. But they didn't sell a lot of those to me. But the Chinese finger puzzle, because the fibers are going obliquely, it allows itself, so the fibers going like this and like this. What it allows is that the space between those fibers can collapse. And so it allows it to squish a long ways down and then stretch a long ways out. And when it stretches, it gets your finger caught. Same way with smooth muscle. So the sarcomeres, if you look zoomed in, they're all arranged like this. Here's, so this would be one sarcomere from there to there. And thick filaments, it's still actin and myosin. Myosin heads, the basic machinery is all going to be the same. It's just that they're arranged a little differently. Instead of Z discs at the end here, we have these called dense bodies, which are, again, just kind of blobs that are proteinaceous anchoring points for the sarcomeres. These dense bodies actually allow some mechanical coupling between cells. So when one pulls, you're going to pull another cell. As it turns out, that'll also cause, we also have gap junctions between cells that allow ion transfer. For example, calcium in particular, which is going to trigger this whole thing, just as it did in skeletal muscle. The upshot of this is that because they're oblique, the cell can really compress. Whereas we can only get about down to 70% of resting length in a skeletal muscle cell. It can only contract maybe this much. A smooth muscle cell can squish down to well under half of its original resting length. So Now, again, the force isn't going to be applied all in a uniform direction, so it wouldn't be as effective in producing force, say, for your bicep to do that one direction force. But it would allow it to contract much, much further. And so we're trading force in one particular direction for work in one particular direction because the change in distance goes a long ways. So work is energy, right? And so if you want to think about the amount of work that a smooth muscle does, it does a lot of work. And as it turns out, it does a lot of work for not much investment in ATP. That is, it's extremely efficient. It burns maybe 20% of the ATP that skeletal muscle would burn for the same amount of work. And that's because it can contract so far. And it also has this what's called a latch state. That is, once the myosin heads bind and drag the sarcomere toward the middle, they'll stay latched and stay holding it without any increase or further use of ATP. Now, exactly how that works, nobody's entirely sure. They're, it's an ongoing bit of science, right? But suffice it to say that they can undergo this latch state. They can squeeze down and stay squeezed and without burning a lot of ATP. That's really important for this is tissue that is tasked with changing, say, the diameter of a blood vessel. Let's say you're out in the cold and you want to decrease blood flow to your skin. Well, here's a blood vessel serving your skin, and it starts out like this, and it needs to turn into this as it restricts the blood going to your skin. So that means the smooth muscle in here has to contract, and that makes the opening, that is where the blood's going to flow, much, much smaller, and the blood will now not flow to your skin. That's a task that's going to last for a long time. If you're out in the cold, those smooth muscles are going to have to go and sit there for hours, days, maybe weeks, who knows? And so it's a good deal that they can do it with very little ATP. Otherwise, you'd be using a ton of it. You'd have all these little titanic contractions going on and cycling ATP, total waste. So that latch state is really important. The finger trap format, what? Isn't this finger trap thing how you describe octopus tentacles? Not yet. Yeah, a similar design in terms of the fibers. That you don't need to know that. Clear. All drawings. High endurance. Oh, the other difference between smooth muscle and skeletal muscle is that the calcium is actually residing outside the smooth muscle cells. Their sarcoplasmic reticulum is small and kind of wimpy. It does have some calcium in it, but it's a lot of the calcium has to come in from the outside of the smooth muscle cell. So that's the other major structural difference. We've been the innervation. So there's a lot going on in this slide, but it had pretty colors, so I went with it. And there's a lot more information that you need to know here. But it has all the working parts. Here's the varicosity, as it's called. These varicosities in the neurons that serve smooth muscle, the varicosities, rather than having terminal ends that contact each cell, you can have a varicosity that has a little like a subway station here, and it can serve this smooth muscle cell. But then it can that same axon can go on down and have another varicosity to another smooth muscle cell a little farther down. So that's that's a difference in the nerves that serve them. But these varicosities are going to release neurotransmitter. Now, depending on the kind of smooth muscle and the organ that the smooth muscle is resident in, there may be different neurotransmitters. In this case, we're looking at the vas deferens. And so this is the spasmic contraction of the smooth muscle and a vas deferens that's going to propel sperm out during ejaculation. And so for that, we have norepinephrine. That's not Nebraska. That's norepinephrine. That's a neurotransmitter. We'll talk about neurotransmitters a little later. And it goes out here and it attaches to a norepinephrine receptor on the smooth muscle cell's plasma membrane, which is the red line there. And when that happens, it will cause ultimately a calcium cascade. Notice that also released by these neurons is ATP, which is also used as a neurotransmitter. Kind of weird, but true. Maybe the first neurotransmitter ever. But anyway, ATP is also released, and that causes also over here, ultimately, these voltage-regulated calcium channels, which are the DHP channels. So again, there's a lot of parts here that are in slightly different configurations, but they're all the same parts. We've seen them all before. They're just tweaked a little bit. And in this case, the dihydropyridine channels are the calcium channels themselves. The change in voltage that occurs here as a result of the ATP causes calcium to enter the cell. So that's where a lot of the calcium is flooding in. Over here, our norepinephrine caused the sarcoplasmic reticulum. INSP is inositol triphosphate. That's just another receptor. You don't need to know that. It's just all the pieces on the picture here. What the R is another ryanodine channel. These are ryanodine channels on the sarcoplasmic reticulum. That causes, just as we saw in the skeletal muscle, a calcium wave that is essentially a calcium action potential. This ryanodine channel opens, calcium comes out, and it opens successively all of the rest of the channels on the sarcoplasmic reticulum, and calcium floods out from the somewhat limited sarcoplasmic reticulum as well. So all of this is calcium into the cell, and the floods into the cell and causes, as it did in skeletal muscle, contraction of the sarcomeres. Actin and myosin are going to chemically interact and shorten sarcomeres. What's the difference between verticosities and synaptic terminals? Functionally, there is none. They're just structurally different neurons. The neurons here, the verticosities, they don't end right here. You can have a series of them. Here's a verticosity, which would be analogous to a terminal end on a skeletal muscle. And then there'll be another one downstream a little farther. It's just a different anatomy of a neuron, different kind of a neuron. And understand, though, that because of that, it's an important functional difference. But imagine this. So you've got an action potential coming down this neuron through this axon. It's going to cause this verticosity to release neurotransmitter. And then a little while later, further down the line, another one will release its neurotransmitter on a subsequent smooth muscle cell. And so while these verticosities are all talking to multiple smooth muscle cells, it's sequential rather than simultaneous. When we saw skeletal muscle cells, we had one neuron coming down, and it would split. So it would serve at the exact same time a whole bunch of skeletal muscle cells, which means they would all contract simultaneously. Smooth muscles, you might have one, serving a skeletal muscle cell right there, and then it'll go on and serve another, excuse me, smooth muscle cell and serve that one, and then it'll go on and serve another smooth muscle. And so this one is always going to be contracting a bit later than this one. Well, that's okay for smooth muscle because as it turns out, it really doesn't matter that much how coordinated they are. It's just sperm in this case. Who cares? If you eventually if you contract that vas deferens, you're going to force all the sperm out. Does it matter that it comes out? Well, maybe it does, all in unison, but certainly not as critically important as coordinating the activity of skeletal muscles. So clear all drawings. What else is there on that? And then we got contraction. Contrast that with this. So notice these words. This is a sympathetic neuron. We'll talk a lot about that in a little bit. The sympathetic neuron is a part of the nervous system, the autonomic nervous system, that is engaged in fighting and flighting. That is, fighting and running away. Fighting and retreating. That's what the sympathetic nervous system does. Fighting, run away, and ejaculating, in this case, are the three tasks. There's more than that, but turns out, at least in male sexual response, so there's one part of the equation. The other part of the equation would be erection. And I put ejaculation before erection. That's unfortunate. Sorry. The parasympathetic neuron this is the neuron that's going to be serving the blood vessels to the penis. So in this case, the same, we got neurotransmitter. Notice the neurotransmitter here is acetylcholine, and ATP is involved too. And we have the same equipment here. We've got our L channels, our DHP channels here. We're going to let calcium in. We have the same calcium spark that's going to cause a calcium action potential in the SR. And in this case, the parasympathetic neuron might cause, in this case, contraction of smooth muscle. And maybe this would be the smooth muscle for the blood vessels that drain the penis. And so if, again, this is your blood vessel, and if you wanted to retain the blood in the cavernous tissue of the penis, you would want to contract these smooth muscle cells in here so they end up looking like this, which keeps the blood in that cavernous tissue, and ta-da, you have erection. So there's smooth muscle in action to coordinate one particular activity, or in the gut, maybe. Could have gone with the gut. That's what I wrote there, for contracting the smooth muscle in the gut. So if you're looking at moving food, you have your gut tissue here, the bagel diagram, and you have some bolus of food in here. And you want to move that bolus along. Well, you need to squeeze that opening down, and you squeeze it down, and the bolus has to move. Turns out there are also muscles, if you look at it sideways, there are also smooth muscles that run lengthwise along the gut. And so between the two of them, you get peristaltic movement. And again, these are not things that have to happen fast. The smooth muscle can kind of go grunch and squeeze and grunch, and these slow peristaltic movements are going to happen. it's far more important that you get big length changes so you can really squeeze this opening down than it is to produce force all at once explosively. You don't want your gut doing explosive things. It's just not good. So anyway, the mechanism for this, again, you have seen versions of the and this is the slide I redrew because the first slide was just stupid. You get these slides free with the textbook. They send you a CD or something, and they give you these slides that some poor new PhD somewhere was paid, 30 cents an hour to put together. And they're most of the time really bad, but they have all the words in there, and it's, I'm lazy. But anyway, I hope this one makes a little more sense, because it should, because it's very, very, very familiar. Calcium enters the cell, it's going to bind to calmodulin, turns on myosin light chain kinase, just as we saw in the skeletal muscle. The only difference is that when the myosin gets, the myosin light chain, regulatory light chain gets phosphorylated, it's required. The myosin heads won't do anything in smooth muscle unless they get phosphorylated. And so it's actually a switch to say, okay, go. Whenever you're ready, go, is what's happening in smooth muscle. So it's a far less discrete on and off. It's just, okay, you're hydrolyzed, you're potentiated, you can go now. And then at that point, the myosin head will hydrolyze some ATP to cock itself, and then it will go through the contraction. So the entry of calcium to the cell is controlling the myosin activity by phosphorylating the light chains. If the calcium levels drop, phosphatase does exactly what it did in skeletal muscles and dephosphorylates it, and the myosin heads go, okay, waiting for the next calcium. The calcium also regulates actin in this way. When there's no calcium inside the cell, there is another protein inside smooth muscle called caldesmin. Caldesmin is bound to actin and blocks the actin binding sites, thus making it analogous to anybody? Making it analogous to, in skeletal muscle, tropomyosin. Exactly, thank you, Prima. And so there's no tropomyosin inside the smooth muscle cells. Instead, we have caldesmin, which is bound to the actin and blocks the binding sites. And so in that case, the smooth muscle will be relaxed. When calcium comes in, it binds to calmodulin, which then binds to caldesmin. When you bind to the caldesmin, it takes it away from the actin binding sites, and the myosin heads can grab on and go through contraction. It turns out that high calcium also causes protein kinase C, protein kinase calcium, to phosphorylate caldesmin and blocks it from binding to actin as well. So you can regulate the actin activity, the actin binding sites, by fiddling with caldesmin. And you can regulate myosin by phosphorylating the myosin light chains. And there's cartoons of all that. This is all you've seen before. The only difference is that calcium is coming from outside the cell here. Didn't even show the SR. You could probably get this happening even without the SR. There's some calcium coming in there, but a lot of it comes from outside. And these are cartoons of, excuse me, both those things, regulating actin over here and myosin over here. Any questions on smooth muscle?
Why did you show both the parasympathetic and the sympathetic on those two diagrams? Was there a difference between the two?
The only difference between the two is really the neurotransmitter that is used. And so in some of those cases, and it would have been better to use here in this one, might have been better if I was talking about vas deferens, or if I was talking about in the case of some smooth muscle, the parasympathetic nervous system you can also get the smooth muscle to relax. So there's mechanisms to actively cause smooth muscle to relax. The only difference in these two slides, though, really is the difference in the neurotransmitter. But I was trying to illustrate that there's kind of two divisions of the sympathetic of the nervous system that control smooth muscle. Smooth muscle is not under conscious control. So skeletal muscle, you're saying go bicep. Smooth muscle, you can't just say go peristalsis. You can't consciously control the smooth muscle that would cause peristalsis. So this part of the nervous system, we'll talk much more about this later. The autonomic nervous system is under unconscious control, and there are kind of two divisions to it. There's this fight or flight sympathetic nervous system that wants action and ejaculation, and the parasympathetic that is more of a rest and digest, as they like to say. Your parasympathetic nervous system is going to dominate when you're, say, moving stuff through your gut. And so essentially these are two examples to illustrate the two divisions of this, a preview of the two divisions of the autonomic nervous system that we're going to talk about here in a little bit. So I'm now going to move on to the slides called neurons, I believe.
One other question.
Yeah.
The changes that are happening in the voltage on the smooth muscle, is that only calcium that's causing that? Or is it the sodium potassium pumps like the skeletal muscle? I kind of missed that.
These are largely calcium-based action potentials. Yeah.
Okay.
Mostly that's calcium. I want to say that there are sodium potassium pumps involved for the entire, if you think about the entire plasma membrane charge, there's always going to be sodium potassium pumps involved. But the real spark, as they like to call it, that initiates everything is a calcium-based spark.
Thank you.
Which is why, and there are a whole suite of drugs, by the way, out there that fiddle with these, L-type channels, and in so doing, fiddle with calcium transport and can thereby fiddle with the smooth muscle contraction. So if you wanted to say a blood pressure drug that would reduce the smooth muscle contraction in the arteries, you could fiddle with these kind of channels and very much change how reactive the smooth muscle is. Anyway, we'll talk more about that later. Stop that one. Whoa. And share that one. Everybody see neurons now? Okay. I kind of glossed over as we were talking about skeletal muscle and the neurons that serve them. I kind of glossed over these neurons and we glossed over some of the details such as action potentials on the sarcolemma of the smooth muscle or the skeletal muscle cells. And so in talking about neurons, we're going to talk much more deliberately and in detail about that action potential that we kind of glossed over on skeletal muscles cells. Now, the same action potential or action potentials in neurons is a bit more sophisticated and has more parts to it because frankly, the distances the action potentials have to travel in a neuron are vast. These are things happening at a molecular scale that have to translate into changes in a cell that might be meters long. And so this is like you're trying to have an event that feels like the scale of me waving or me shooting an arrow, and it's shooting an arrow toward Uranus, and somehow I've got to affect what's happening on Uranus or Pluto, and it's a really long ways away, and all I've done is shoot an arrow 200 yards. And that's what's going on with neurons. We have very much molecular scale things happening, events that are driving macroscopic, very macroscopic events. It's quite remarkable. So neurons have really weird, if you thought skeletal muscle cells were weird, neurons are equally bizarre. Some of their peculiarities here, neurons are amitotic, that is kind of like skeletal muscle cells. Once they're grown up, they can't make copies of themselves. It's kind of a bummer. You kill a neuron, it stays dead. If a neuron feels the need to do more, it just can't make more babies. It's done. You have the neurons that you're gonna have right now. So now, what you can do with them and how they interact, they can grow and they can make new connections, and that's the key. They have extremely high metabolic rates. Remember all those sodium potassium pumps that we saw all over the sarcolemma of skeletal muscle cells? Yeah, you got all that happening on neurons as well. Every bit of them, it's just covered. Their plasma membranes are studded with sodium potassium pumps. I don't know, somebody must have done this calculation, but if you think about the ATP, the energy that your body uses in a given moment, and how much of that energy is taken by one particular mechanism, sodium potassium pumps, between sodium potassium pumps and myosin ATPase is when you're moving, that is when you're going for a run or something, then there's going to be a lot of that energy in myosin ATPases as they hydrolyze ATP. But even as you're sitting there now doing skeletal muscle wise, not much. The neurons, hopefully, are still using buttloads of ATP as they drive sodium potassium pumps to create membrane potentials all over the plasma membranes of all of these neurons. And you have billions of them. It's an enormous investment. This pile of neurons up here is a spectacular investment. 20% of your basal metabolic rate as you're sitting there right now, just right there. And all of that is, not all of it, but most of it is just sodium potassium pumps. So basically, you got two biochemical events going on inside a vertebrate body, sodium potassium pumps and myosin ATPases that are probably taking 90% of your energy. Two little molecular motors, machines, just running all the time. Neurons have to be long-lived because they're not mitotic. They have to live a long time. So that's a difficult thing for a cell to do. You have to have a high metabolic rate, which means you can create all those reactive oxygen species that tend to oxidize things you don't want oxidized. And so high metabolic rate is generally not something you want when you want something to live a long time, but neurons manage to pull it off. Exactly how long could they live? That's a good question. it's pretty easy to imagine fixing things like organs like hearts. And grow a new liver, grow a new kidney. You can imagine fixing a lot of these organs, which are largely mechanical in nature. But
Me, sir.
Excuse me? Don't know what that was. Was that a chat? Sorry. Here we go. Whoops, lost my cursor. There we go. However, if you're thinking about immortality, coming up with a way to make sure that neurons don't die is there's an interesting question. Now, could they live a really long time? Presumably, if you made everything else work well and they continue to serve neurons well, your cardiovascular system continued to serve neurons well, could they live forever? I just don't know. It's difficult to imagine. Obviously, they can live a really long time. There are turtles out there that have neurons that are 200 years old. There are whales out there that have neurons that are probably 200 years old as well. So it can happen. We'll talk more about that later. They're weird. They're so weird that all of their cell components tend to get special names. For example, the rough endoplasmic reticulum, where protein synthesis happens, those are called missile bodies. They have a ton of mitochondria, predictably, because they use a lot of ATP. They have a very extensive cytoskeleton in the form of microtubules and neurofibrils. These are the interstate highway system, or perhaps better, the railroad of the cell. A very important railroad, because this is a cell that, again, is microscopic, molecular in its machinery, and at the same time has to translate that over enormous macroscopic distances, meters and meters. And so there's a very effective and efficient railway system inside the neurons to move organelles. Whole organelles get moved, proteins get moved, all kinds of material goods get moved on this railroad along these microtubules. We'll see them in a second. Some nomenclature here. The cell bodies of a neuron where you have clusters of neurons in their cell bodies, we call them nuclei. In the central nervous system, that's CNS, central nervous system. That's the brain and spinal cord. The peripheral nervous system has collections of cell bodies, and they're called ganglia. So you'll see those terms again. Lost the cursor. There it is. So the general anatomy again here, there are going to be cell bodies, and then there's going to be processes sticking off of these things. So you might have a cell body here that, a cell body, and then it has a process sticking way off of it like this that goes for a really long ways. And here's the cell body. And then you might have processes at this end too. And I've drawn a Nereocystis, the bullwhip kelp. Anyway, the ones at one end, which would be the input end of a neuron, that's the dendrites. And these processes, usually just one long one that can then split into many later, are the axons going somewhere. That's the output end. Both of these sections, the dendrites and axons, are going to have resting membrane potentials that can be triggered into carrying potentials to other parts of the cell. They differ slightly in how they propagate, and that is the ones at the dendrite end are called graded potentials versus the ones at on the axon end are action potentials. Once you get down to the end, at the end of the axons, we have, as we saw with the skeletal muscle, we can have many, many terminal branches, thousands or tens of thousands for a single axon, which I've never tried to draw. But imagine one axon coming down and splitting into 10,000 little ends, all of which will talk to, say, a skeletal muscle cell, 10,000 of them. So that means for one action potential coming down the axon from one neuron, you can affect 10,000 things out there. They have little knobby ends that are full of neurotransmitter, and we've seen some of those in action already. Far better picture. And this is a typical face of a neuron, and it's typical, as many things are typical when we draw typical things. They're easy to draw, but they really don't reflect many actual neurons that look like this. So the neurons only interact with. No, neurons act, there's all kinds of stuff that neurons will interact with, all kinds of tissues, and we'll see a variety of them, but they can interact with muscle, they can interact with other neurons, they can interact with tissues that produce hormones, for example, and excrete things. So they can talk to a lot of different kinds of tissues. Anyway, this is a neuron. This is a typical one, but it doesn't look like any neuron you'll probably ever see. And it looks more like an alien with a single leg, or an alien. What it looks like is an alien with really wild hair, one eye, and a very thin body with 6 legs at the end. There's 5 legs. So this is the axon part of the neuron. This is the body or soma. These are the dendrites. Here's an actual neuron. And notice that, look at all of the projections coming off this neuron to talk to, it's messy. It's talking to lots of other probably neurons in this case, which is why, nobody ever tries to actually draw a real one, because the real ones are messy. This is no doubt a neuron probably in brain tissue. So to give you an idea of the complexity, the number of ways that you can wire together neurons. That way is the number of pathways. A neuron over here could talk to this neuron over here. It may be that pathway is blocked, but you could go this way. It's like a road system, a really complicated one. It's like Boston. If you've ever tried to drive in Boston, don't. Just take the subway. Driving is just not going to happen. New York, same thing. It's just no. There's lots of ways to get there. The number of connections, the number of ways you can put together neurons in a human brain, frankly, any vertebrate brain, is unimaginably large number. This little railroad system that goes on, they call when you're sending products out down the axon to the terminal end, they call that anterograde versus retrograde is coming back toward the nucleus. And as in all cells, the nucleus is going to be where actual transcription, translation happen. That's where we're going to actually make proteins that are involved in the business of being a neuron. And again, a lot of what goes on in a neuron happens down here at the terminal end. And you're literally, in this case of this neuron, you're making goods that you're going to have to transport to Saturn and get them there in some kind of timely fashion. And it's a really long ways away. And you're going to have to carry it with a Prius. Not a rocket ship, not a Starship Enterprise, but a Prius. It's going to be slow. Things are going to be moving at a molecular scale. Here are the 2 molecular scales at which they move. You have, so anterograde going this way, retrograde going back the other way. And turns out there are two different proteins that are molecular motors. And these molecular motors, they're called dynein and kinesin. And they're not really represented on here at all. No, not shown. But notice that the size of the retrograde here, the dynein, is really big. These are molecular motors. These are exactly like analogous to the myosin heads that we saw in skeletal muscle. And these myosin heads in this case are going to be, rather than moving the thin filaments, what's going to happen here is the myosin head, or not myosin, but dynein heads are going to, instead of moving something, they're going to move and walk along the railroad in the form of these microtubules, so these proteins that form the cytoskeleton of the neuron. And so they can walk this direction, retrograde. Turns out this is a bit faster than anterograde. The retrograde has bigger step motor molecules. Exactly why? I don't know. But anyway, the movement of stuff from the ends, terminal ends, back to the cell body, this is the recycling portion. So all of that went on in the synaptic end, and we'll see a whole bunch of different proteins here in a little bit. But all of these proteins have to be made up here in the cell body. When they get used, the protein, the amino acids, are going to be recycled. You just don't throw that kind of stuff out. This is exquisite recycling. And the step to take the components back to the cell body where they can find a rough ER, the Nissl bodies that will put them back together, that step is relatively fast compared to the supply line out to the ends. If you want to think about it, why would it be better to make this a bit faster than this going down? Why do you want this recycling to maybe carry stuff away from the end at a faster rate than you can carry stuff down to the end?
So you can constantly replenish.
Well, you got to constantly replenish is the key here. But there's a disparity in the rate. There's a disparity in the rate that we're carrying stuff away from the end versus down to the end.
Isn't it because if you have a higher rate of stuff being carried to the end, you would eventually just run out of stuff to actually transmit it?
Well, you're carrying stuff, you carry stuff down to the end, and so you're carrying proteins down there that get used, right? So what happens, and here's the analogy back to New York City. I pick on them a lot, but how can you not? If you were to imagine the goods going out to the tenants of New York City, and they're eating them up, right? They're eating this stuff, using it at a certain rate, and there's that rate of sending it in to the homes and apartments. But then there's the rate of taking everything out, the garbage out. What happens when the rate of coming into the homes exceeds the rate of getting rid of the garbage? Do you want to live in that apartment? I did. When I was about your age, I think that's probably what our place looked like, the garbage tends to accumulate. And pretty soon the garbage accumulates to the degree that you can't get anything done. And you need to take out the garbage. So I suppose it would be bad if this were rate limiting in the sense that the neuron can't function as well because it doesn't have the components down there it needs. But even worse is if you pile the stuff up at the terminal end and you can't get rid of the garbage that you've created, you can cause much, much larger problems that ultimately may affect the viability of the neuron. That is, it might get damaged. And since they have to last forever, you have to be really assiduous about your recycling. And They have this lovely little railroad system that's going on in there and constantly moving things back and forth. Incidentally, viruses in particular love hitching a ride on this railroad. They're the hobos of, that's probably politically incorrect to call them hobos, but that's what they are, of the neuronal world. Viruses such as herpes viruses, rabies, love hitching a ride on these railroads, and they get carried as a result. Those viruses couldn't do anything in terms of replication down here at the terminal end because there's no machinery down there to make proteins. They have to get up here to the cell body to actually make copies. And so they hitch a ride on the railroad and jump a railroad car, and they end up back in the cell body, and they then can undergo replication. Well, there's more pictures of them, kinesin and Dyson, or Dynan. Anyway, any questions on that so far? The structure, basic structure of neurons?
Yeah, so is Dynan only used in retrograde and kinesin only used
Yeah,
in
they have
retrograde?
this, yeah, right, exactly. They have and Dynan is the one that it's a bigger motor molecule, and so it's geared differently. It takes bigger steps, if you will.
Okay.
Is that the reason that there's two different kinds? That may be the selective reason that there's two different kinds. So you have a disparity in rate of transport, and so you have always making sure that the terminal end is clean down there and works properly. That's a guess. So on to, that's the machinery. Not mentioned were all of the sodium, sorry, question?
Yeah, do you have, how does the Dynan get down to the end to then bring back whatever garbage it needs to? Like, do you have Dynan and kinesin also transporting each other?
Oh, great question. Yeah, empty rail cars. I've never even thought about it. What a great question. I would imagine that, let's see, would the Dynan then jump onto the kinesin and get carried back, and do they cooperate that way? I cannot remember. I don't know if I've ever read that. You can look that one up. That's a great question, though. Obviously, yeah, you need to move. Is it only one direction? Not sure that I can't remember now. What is the mechanism? I did read that once, and how they change tracks, essentially. Now I can't remember. I'll look that up and get back to you. Yeah.
Our microtubules are constantly being like formed and then broken down, right? So do these Dynan and kinesin molecules ever detach themselves from those microtubule subunits?
Yeah, they do. Yeah. But I'm not sure that's the one that's routinely used, because when you're talking about, I'm thinking about really long axons now. And so in those cases, microtubules, that kind of cytoskeleton is quite a bit more stable than, say, the cytoskeleton in the dendritic end, where we're changing. And certainly when you're in a place like the brain where the neurons are really close together and the axons are quite short, that can be a much more dynamic process. I'm naturally thinking of these really long neurons that are so extraordinary in their structure that they have to move things meters. And in that case, the cytoskeleton is a much more stable thing. But yeah, I'll look that up. I'll read up a little more on that. But I can't remember right now. That's a really good question. Left out of all of that description of what's going on was the plasma membrane, because we saw a lot of the plasma membrane in the skeletal muscle cells. And just like skeletal muscle cells, we have a membrane that is studded with ion channels, sodium channels, potassium channels, sodium, potassium, ATP pumps, ATP-driven pumps, and resting membrane potentials. And so we're going to be looking at the way these membrane potentials change on an axon as you move along the axon. And we're going to be looking at graphs that look like this. So this is on this axis, membrane potential in just millivolts. And then this is just time on this axis. And so if you looked at, here's our minus 70 resting membrane potential on the neuron plasma membrane. And when that voltage would increase, that is from minus 70 to minus 60 or whatever that is, that would be an act of depolarization. That is, it's not, I don't like the term, but this is, we're stuck with it. It's depolarizing in the sense that it's getting less polarized. It's getting closer to zero. Zero would be exactly depolarized, right? So this is a process of depolarization, but it is not to say that the plasma membrane is completely depolarized, right? Because it's not zero, it's still minus 60. Contrast that with terminology given to this event, which is hyperpolarization. That is, it's getting more polarized than it normally is. So it's down to minus 80 or whatever that is. Okay, that's the difference between a depolarizing event and a hyperpolarizing event graphically shown. So looking at the dendritic end, we're going to be looking at these depolarizing events at the dendritic end. That's the input end of neuron. That is, this is a neuron being spoken to by another neuron. And by whatever means, and we'll talk about the various means here in a minute. It's kind of a cycle of one neuron getting an input and sending it on to the next one and sending an output to the next one. And at the input end, that is the dendritic end of this neuron, the degree of depolarization, the magnitude of this depolarization, that is, did it go from minus 70 to 0? Did it go from minus 70 to plus something? Or did it just go a little bit? The magnitude of that depolarization is going to be proportional to the strength of the input stimulus. The analogy I think that is intuitive to most people is it's the difference between a little touch as far as relatable events that your neurons perceive, a little touch, or is it a ball-peen hammer? Obviously, the ball-peen hammer strike is going to be a bigger stimulus than the little touch. And so the dendrites of the neurons that perceived that will be changing in their membrane potentials proportional to the strength of the stimulus. So here's what's happening at the dendritic end. So let's say we had some, in this case, we have, again, sodium-potassium pumps that are driving sodium outside, potassium on the inside, and we have a relatively positive outside compared to the inside, minus 70 volts, volts, that would be a lot, millivolts. And it turns out, and this particular neuron is a neuron that has at its dendritic end, such as you would on those neurons and the neurons in your face that detect this touch. Those neurons have what are called mechanically gated sodium channels. That is, these are sodium channels that when you push on the neuron and it deforms the plasma membrane, it causes those sodium channels to open. And then sodium rushes in. And as the sodium rushes in, it will spread. Again, there's relative negativity on either side of this, right? So from the point of this stimulus here, we had sodium rushing in and sodium spread laterally. The amount of sodium that came in is going to be proportional to the number of sodium channels and how perturbed they were by that input stimulus. Was it a touch? They weren't perturbed much, very little sodium came in. Was it a ball peen hammer? A lot of sodium channels opened, a lot of sodium rushed in. The more of that sodium that rushes in, the farther it will spread laterally from this, or perhaps more properly longitudinally down this dendrite. It would go both directions, right? Because it's relatively negative in both directions. And the positive ion sodiums are going to flow toward the negativity. Okay?
So essentially the mechanism to open these channels is it's kind of like a ripple. So like if I threw a pebble into a lake, tiny ripple, not a lot of channels are opening if the lake is analogous to a neuron. But then if I threw like a hunk of concrete in there, I'm getting a lot of ripples, so a lot of channels are opening.
Yes. I mean, a pebble versus the hunk of concrete is a good analogy. Absolutely. The ripple itself, perhaps not, in the sense that we have water molecules moving other energy passing through these water molecules. But yeah, you're right. That's you can think of it that way. This is going to be this one's a big splash. The difference, well, and similar to that, I suppose, in a way, that the wave eventually fades because it loses energy. In this case, this wave of sodium ions is going to decrease in its strength as it moves away from the point of stimulus. That is, it's decremental. So in other words, if you were to measure the voltage change at any point along that dendrite, you would find the further you get away, the less voltage change on the plasma membrane you'll see. So, in other words, if this right here was the place where you hit it with the ball peen hammer, so this is distance now, and this is membrane potential voltage, started out at it's minus 70 over here, minus 70 over here. You hit it with the ball peen hammer right there. And so that voltage, a lot of sodium rushed in there. But as you tried to measure that change in voltage, if by the time you got, say, a few millimeters away, you would find that there was no change at all. That is, it's the sodium may rush in, but
you kind of just became a robot.
Yeah, same for me.
Same here.
Sorry, you're all cut out. I paused for a second.
Yeah, we lost you there for a second.
Okay, everybody back?
I think so.
You're back now.
Kinda?
Well, Kelsey, you're the host now. Would you like to teach us about neurons?
Not particularly.
All right, well, thought we'd give it a try.
Yo, what? I can press the record button.
Does anybody know what's superficial to the plasma membrane of the neuron? Like the fluid that it sits in, or does it at all? Like, where is the sodium even stored? Does anybody know?
Sorry, I was trying to diagnose what happened there. No idea. Solar flare, electromagnetic pulse. We're all going to die. Where was I? Share screen. When did I cut out?
You were mentioning the decremental nature of the
Okay.
neuron.
Yeah, at least
Riley had a really good question while you were gone.
Who did?
Hey,
Riley?
Professor
Doesn't
Ward.
matter who.
Where is the sodium stored? Like, what is superficial to the plasma membrane of the neuron? Like, does it?
Oh, so why doesn't the sodium just leak away into space, kind of thing?
Right.
Yeah, all of these structures. So in the case of the skeletal muscle cells, we had that connective tissue sheath over the top, the endomysium. As it turns out, any of these collagen-based things are really good at insulating and holding charged ions. As it turns out, in neurons, we also are going to see some very specialized structures that essentially hold those ion channels very locally. One of the things that's going on that you tend not to think about is that when there's always tissue around the outside, and so the ability to diffuse things away into space. If you had an axon, even in a petri dish of fluid, it would continue to pump sodium ions in the same way. Eventually, it would diffuse out away, but these are such small scale. The molecules themselves are such small scale relative to the size of the plasma membrane, which is enormous. In all of these cells, these are really big structures. And so the ability of atoms to move away after just being pumped across is somewhat limited just by their scale. You add on layers of tissue, and there's really nowhere to go. And we'll talk more about some of those neuron whole scale things in a little bit. Anyway, yeah. It's decremental. So it's decremental in that, so if, again, here's where you stimulated the plasma membrane. If let me do this a different way. So this is, let's say, minus 50. I don't know what it is. And that's with the tiny, gentle touch. Hit it with a ball peen hammer, and you might see it go like this. Actually, this can go either direction, right? So should draw like this. You hit it with a ball peen hammer, and now it reaches this. It's still decremental in the same exponential decay, but at the same distance out here, on this small touch, we saw no change in voltage this far away, say 2 millimeters, I don't know. But 2 millimeters away for the ball peen hammer, and we see, ooh, it's still up at minus 50 on that one. And so if you then extended this further, it might be, 5 millimeters away before you saw zero voltage again change from that one event. We're all drawings. That's illustrated in probably much better in this picture. So if here was the event, in this case, our stimulus is coming from another neuron, and this neuron says, go, go, gadget dendrite. And here, the voltage change at that point is up to minus 40, but a little farther away, it's only up to minus 55. And when you get all the way down to here, it's barely noticeable. Now, in some cases, that will mean that the neuron, the receiving end neuron, will not detect it at all. In other words, there'll be no response. The whole point here is to get the receiving neuron to respond in some way. That is, I'm sending a message. I want you to send this message on to, say, a muscle cell. In this case, we have a cell that is receiving a stimulus. It was this big right where it happened at the dendrite end, but because of the leaky plasma membrane, and this is a decremental graded potential, turns out by the time you got a millimeter or so away, the voltage was so low that it turns out that the neuron didn't do anything. And the reason it didn't do anything was that this part of the plasma membrane, here called the trigger zone, in this trigger zone, there are further sodium channels that can only be opened by voltage. That is, they are voltage, let's clear no drawings. They are voltage regulated sodium channels in this trigger zone. And the voltage they require to open has some threshold. Here, they're saying it's minus 55, which is a pretty typical number. In other words, if this potential here at the graded end had been, say, a little bigger, then it would have been quite a bit bigger all the way down here. And by the time, even though it had lost its voltage to a certain degree, by the time it got to the trigger zone, it was still above this threshold to open the voltage regulated sodium channels. And as a result, if we had a bigger graded potential here, we would end up with a voltage that was above the threshold down here, and those voltage regulated sodium channels would open. In that case, the neuron is going to respond. But in the original case, as drawn, this input was insufficient. The graded potential pooped out before it got to the trigger zone, so nothing happened. The neuron didn't respond.
So if the threshold of the voltage regulated sodium channels in the trigger zone is negative 55 volts, does that mean the stimulus must be greater than negative 55 in order to reach the trigger zone in time?
Right. Well, yeah, obviously because it's decremental and it's some distance away, it's going to have to be more than 55 up here for it to end up at just 55 down here, this far away. So it has to give you at least 55 in this trigger zone for these sodium channels to open. So if it were only 55 up here, there's no way for it to be 55 that far away. So that means if it's minus 55 here, it's going to have to be something quite a bit more positive up here for that graded potential to maintain at least a minus 55 voltage all the way down to here. Was that kind of your question?
Yeah, thank you.
I mean, just like there's a voltage that these proteins are just not going to, they have to see a certain voltage for their conformation to change. When that happens, and this will be the last thing, when that happens, so we have voltage regulated, those voltage gated channels right there in the trigger zone. Turns out there are voltage gated channels, just like we saw with the skeletal muscle, all along this axon. The first voltage gated sodium channels are in the trigger zone, but they are the same voltage gated channels all the way down the axon. And so once the sodium rushes in here through those voltage gated channels, it will spread this way and trigger other voltage gated channels. Just as we had in the sarcolemma of the skeletal muscle cell, we will have an action potential that will self-propagate down the axon as each subsequent voltage gated sodium channel gets opened by the change in membrane potential. But that can't get started until this trigger zone gets a sufficient voltage to start the whole thing. All righty, we'll stop there, and we'll finish up neurons on Thursday. And I'll go over the exam on Thursday recitation. If you have questions about your exam, and you should, by the way, look at your exam. Many of you have already emailed me finding mistakes in the grading. I had the computer trying to look, and then I had to correct it to grade these things. And I'm sure I missed some. Well, I know I did, because you've already told me I missed them. Anyway, stop share. Any questions before you leave?
I had one.
Oh,
sorry.
No, go ahead. Mine's for later.
I just had one quick question regarding the signal traveling along the axon. So yeah. I took a psychology class years ago, and I'm trying to remember the function of the myelin sheath. Was that to regulate the speed at which the signal can travel?
Not to regulate it, it's just allows it to travel a great distance. And I'll get to the myelin sheath in a little bit. I have to say, a cautionary tale there on psychology textbooks. When I was first teaching, this was a stunning revelation to me. I was first teaching A&P many, many years ago. And presumably they've changed psychology textbooks since then. But a student came up to me after this, essentially the same lecture here, the same content, and asked me, he was just terribly confused. He walked up with his psychology textbook and he said, I'm confused. I'm trying to wrap my head around what you've just told me and what's in this psychology textbook. And I, well, let me see it. And they had a description of how action potential is propagated in neurons, and it wasn't even close. And I just said, I think I understand your confusion. Go get your money back on this textbook because it's a piece of crap. So hopefully you have a newer edition of some psychology textbook than that, and a better content. But the myelin sheath is what allows fast propagation of action potentials over great distances, over meters. Otherwise, we would have no hope of coordinating neuronal activity over macroscopic distances of vertebrates unless you have myelin sheaths. If you ever want to see what happens to myelin sheath, to our neuronal activity with no myelin sheaths, watch someone with multiple sclerosis try to move. It isn't pretty. That's what happens when you don't have a myelin sheath. The neurons can't propagate action potentials, and they can't coordinate their activity. But I'll talk about it. I'll talk about it on Thursday too.
Okay, thank you.
Yeah. Elena, do you have a question? I think it was your Hollywood Square that was lighting up. I don't remember.
It's more of like a series of questions / statements.
All right.
That doesn't have to do with this class. That's why I was waiting till the end.
Oh, okay. Okay. Well, you can ask me in private too, if that's what you want to do. I don't care.
I mean, it doesn't bother
Anybody
me.
Other have, like... Any other more related content, not series? No?
I have a quick question.
Yeah, Sydney.
On the slide that's the mechanically chemically gated sodium channels, I missed it. You said that when sodium rushes in, that it's proportional to something, but I didn't catch what you said.
Well, the strength of the stimulus. So in other words, if you hit the neuron with a ball peen hammer, it's going to open a lot of sodium channels, and a lot of sodium will rush in, which means that that voltage change is going to be bigger and felt farther away from the point of stimulus than if you just gave it a very light touch.
Right. Okay. Perfect. Thank you so much. Have a good day.
Yep. Yeah, you too. Sun's out, kind of. I have a question for verbatim captions. Are you there? I don't know what verbatim. Are they just? Is it artificial intelligence that is transcribing everything I say? That would be an interesting transcript to read.
It's the government following you around, clearly.
Yeah. Well, I would like to see how they verbatim captions captures phosphorylation, for example. What word actually appears when I say phosphorylation? It's probably porpoise or something. Who knows?
It might just say like photosynthesis or something.
Probably photosynthesis is the pH word that just automatically happens.
Why would the government need a plant in a Zoom call? They already have birds to watch us.
Yeah. No, they don't.
Yeah, they're government drones.
Yeah, no, they're government drones, yeah, but
the birds are government drones.
Oh, I see. Birds are government drones. Somebody approached me about doing research on using birds as drones, as you suggest, and training birds and actually creating cybernetic birds to that you could control, because they're so much better flyers and they don't have this battery problem that drones have. I think I was less than gracious for their interest. I think I may have laughed at them and said, nah, fuck no, not doing that. But anyway, yeah, take that, verbatim captions.
That's actually kind of an interesting, like, I guess it's not really a story, just more like well, I guess it could be called a story, but that's kind of interesting that was a real project, and I'm just talking about this stupid joke theory.
Oh, that is out there. What was this, a QAnon thing that they say there are bird drones out there taking pictures?
I think the bird drone thing. I think that was like before the whole Q thing. Pretty sure the birds are government drones thing. I'm pretty sure that's older. I just know it's a stupid theory that people share for fun.
Yeah. Well, I mean, in reality, I've also been approached because I did hummingbird research. There are folks out there that have created little hummingbird-like drones, and they look a lot like hummingbirds, and they can fly kind of like hummingbirds, and they're very useful platforms for small cameras, and you can fly them inside a building. And they've actually used stuff like that in terrorist hostage situations and so on and so forth, to get pictures on the inside. And presumably that allows, if it's just a hummingbird in the building, the terrorists won't shoot it like they would shoot a regular drone, right?
Mm-hmm.
But at the same time, now you're saying that any bird that flies close to a war zone is going to get shot because it's assumed to be a drone. So that right there was why I simply refused to even consider it, which is, now we're dragging another species into our wars. Isn't it bad enough that we destroy all their habitat with wars? I'm not going to actually engage them in the conflict. So I declined on that research avenue. By the way, it's very profitable research avenue. The Defense Department has deep pockets and funds some really interesting stuff. Many of my colleagues have been funded by the Defense Department, and it's not all malevolent stuff. It's some of it's just kind of interesting. Yeah, it has military applications, but it's not nasty. But yeah, not interested in drones. If I see drones, I reach for the shotgun every time.
It's always a fun time in this class, isn't it?
Yeah, verbatim captions. Who knows? I'm probably going to end up, yeah, who knows, before a grand jury somewhere. Elena, do you want to log off and go to another chat, go to another venue?
We can. I mean, I also don't really care. It's not like it's top secret info or anything.
Okay.